first I want to thank Josh Eccles for inviting me to come talk with you all today about what our partnership is doing to develop. At the end of the day, I hope that you kind of just take home how LabKey is accelerating biomarker development. But first I'll start the talk and kind of tell you a little bit about what I do at Genentech and what our group does, and kind of then set up kind of where LabKey comes into it. And then we'll step back in time a bit, and then we'll kind of get to how I figured out I needed a solution, and where we are now and where we're going, our vision for where we're going. So I'm in a biomarker development group at Genentech, and we support the development of therapeutics throughout the entire pipeline, so all the way from research all the way into clinical development. And the biomarker development pipeline has different focuses kind of depending on where the molecule is along the pipeline. And kind of at the front end, we're focusing more on finding biomarkers that will tell us that our drug is hitting its target and the mechanism of action, and help us find the right dose for our patients. And so those are the pharmacodynamic biomarkers. And then we're shifting through, and our focus is changing more to trying to find predictive and prognostic biomarkers. So biomarkers that will help us say, are we getting the right drug to the right patient? And so one of the tools, and the major tool, and my technical background is in mass spectrometry. And so this is one tool that they brought me into Genentech about 6 years ago to bring this technology to our group. And we're you can use it throughout the biomarker development pipeline, either more on the discovery side that can be targeted or unbiased, or more in a focused manner where you're really kind of testing hypotheses and narrowing down on which biomarkers that you want, and over here probably having really robust quantitative methods that might even be diagnostic methods. And so here's where LabKey has come into our lives, is where it's helping us kind of on the data management piece. And I'm going to talk to you specifically about the Panorama piece. But one of the nice features is how customizable, and we've already been hearing some nice stories about that this morning. So again, when I came in, the vision really of our director was, I really want you to bring mass spec in and kind of demonstrate to our group how we can accelerate biomarkers using mass spec, because we do all of these discovery experiments and we generate these whole host of candidate biomarkers through sequencing data or through the literature or some exploratory studies or target-specific biomarkers. We have hundreds of biomarkers, and how are we going to, in some of these studies that we're doing, figure out which ones we want to get down? Because for those of you who might be familiar with like immunoassays, it can take a lot of time to develop the antibodies or reagents that you need to develop those assays, whereas mass spec, it's relatively quick, at least for some of the protein biomarker work. So I started developing some targeted assays that were looking at some multiplexed assays to look at these biomarkers and try and help us more quickly figure out which ones are going to show a response in response to treatment. And my, it says CSF here, cerebral spinal fluid, because in particular I've been supporting Alzheimer's biomarkers, so that's my background, is neurodegenerative disease. And so in this I had a whole host of, say, like 50 proteins that I wanted to go after. And though this is maybe a little more, it's a high-level overview for more of a mass spec-based audience, I just kind of was going to walk through this just to give you a sense of the complexity of our types of data. So we take a list of proteins, but then we break them down and pick signature peptides for those proteins. We then want to have standards for those peptides, so we have stable isotope standards that we use for the quantitation piece. So we're now kind of already duplicating our efforts here. And so then within those peptides, we're picking what we call transitions, or like specific fingerprints for these guys, and we do multiples of those. So now we're like amplifying this one thing into like 8 different things, say, that we're monitoring for that. And we sum the area under the curve for the sum of the transitions for that. We do the ratio of the internal standard, the heavy peptide to the light. We do this in a multiplexed manner. And so you can imagine this nice, pretty picture, but then underneath it all, there's a lot of different data to kind of be keeping track of and QCing. So a software that was developed right here in Seattle, and the lead software developer, Brendan McLean, is here at the UW, is Skyline. And so this software was initially developed and has now expanded to support a number of different mass spec approaches, but was what I started using to help me do my method development and refinement for this. And one of the big key features of why we turned to this was because it's vendor neutral. So that was really nice. We had different instrument types in the lab, and so it was really beneficial to us. And this was just kind of a quick snapshot of some of the data where I had like all of my peptides that I had in the method, but I could quickly like click on a peptide and get a view of, this was an example of where I was looking at how a calibration curve and some QC samples were performing, and so the internal standard looked relatively the same across. I could quickly look at the peak shape and look if the peptides were coming off at the same time in all of the samples. So it was kind of a nice software tool for that. But now here's the piece where I'm going to kind of step back in time, and which we already had a nice preview in the last talk, that they speak about Excel files, you know. So I was developing the method in Skyline, but then I'm generating the data and have the vendor software that's helping me do like the peak integration and the cal curves. Then I'm doing copy pasting into Excel and trying to figure out kind of manually for all of the peptides and all the transitions more manually how to QC this. And this is great and fine when you're in academia and you're a postdoc and stuff, but when you're in industry and they really want you quickly to be able to make a decision and quickly do things, this isn't going to be sustainable, right? So this ended up being like a very iterative manual development. And I'm trying to like prove myself to my group that mass spec has value, and they, you know, are saying that it's going to take me like months to get through the data. So that's not going to be really cool, right? Maybe it took me 2 weeks to develop the method, but I can't even analyze any of the data from the data set. So we really needed to find like kind of a solution to make this whole process simpler and more automated. And the other important piece is the trackable nature, right? Like I need this long term to be a robust method for clinical samples. And if I leave the company next year, which I'm not planning to do, but if something happens, right, someone else needs to come along and be able to figure out what did I do, and, you know, in a trackable manner, trust the data. So I went to a mass spec conference a few years ago, the big one, and this kind of piqued my interest. I was like, okay, LabKey has developed, what is this Panorama? And so it was a module that was developed to help with the data management piece for Skyline specifically, Skyline specific data. And I'm like, okay, this piqued my interest. Tell me more. I want to learn some more. So how are other people using this? So it turns out that this was a public, Skyline's very open source, it's public, and so is Panorama now. And so a number of folks are starting to use this and to kind of be able to share their method files, and even journals are asking us to put our data here so they can actually take a look at the raw data that's going into the figures that when you're submitting publications. So this is pretty exciting. So I wanted to know a little bit more. And so one nice example of this was a CPTAC, which is a clinical cancer proteomics group. So this was their kind of, you can't really see it very clearly, but they're showing some visualizations here of their chromatograms as well as their response curves and pulling from their Panorama server. And so this kind of really piqued my interest. And we'll come back to this piece of, you know, actually kind of doing the data processing and visualizing within LabKey. But I was like, well, this is all really public. So how am I going to do this at Genentech? Right? So then we started talking to Josh and Brendan, and we said, well, what about you joining the partnership, this Panorama Partnership? And so it was an opportunity for us to basically get our own server on site and set it up so we could have it within our firewall in a secure system, and that we could kind of work together, because it's kind of a living, breathing thing. That's part of what we're all here today to learn from each other and how we're using this, but is to kind of guide the development priorities and to suit our needs and customize it to what we were doing. Basically, LabKey and Panorama and Skyline have really grown. And because of the kind of adoption throughout the field on this, there's a lot of people that are supporting this, including us now. So it's been a fantastic journey. So one of the first things that I was able to take advantage of right away was kind of being able to build some libraries within Panorama, and then others within my group could start saying, well, did I already develop an assay for this peptide? And so I even have my colleagues that are over in Europe, our parent company Roche, they were screening like 8,000 peptides. So now, they're able to put this, and we're able to share these libraries and more quickly develop and download the methods. And so then kind of the next piece, kind of that vision statement where we were kind of figuring, well, we want to not only be able to archive our data, but we also want to be able to kind of look at it within the server and get a sense of how things are going. And this was kind of, again, from the CPTAC page where we got this idea of, well, we could actually, run these scripts and do these visualizations right within LabKey. So I'm going to tell you a bit about a few of the features that we've been developing through the partnership over the past few years now. So one thing that's been cool, Skyline, you do a lot of kind of method refinement where you narrow down on what peptides and which transitions you're going to do. And so now one of the features is we can kind of link those methods together, and you can kind of quickly view, oh, here's like the most recent method and how many proteins and things we're looking at, and you can quickly download the most recent one. But you can also, with this view, kind of track what's changed over the course, like what's dropped out. So that was kind of a nice way in order for us to track our method development. But one of the biggest features that we're taking advantage of and one of the first projects that we kind of wanted was, what does every instrument kind of need to do, is kind of more this system suitability thing. You want to walk up and kind of know what's the status of your instrument. So we built this new QC folder type, and I'm going to go into more details, but basically it's kind of a way in which that we can now upload our data and take a look at performance of standards over time. And so one of the features, for example, here, and the gray is not really showing up, but basically you can define, for example, this window, say these 10 samples, and create what we call a guide set. And you say, run the statistics against those guys. Those guys are how the instrument should be performing. And so we can kind of quickly take a look and see when things kind of jump out of spec. And we can do that across peptides now, so we can figure out, is it just one peptide that's an outlier, or is there something systematic that's going on that we need to kind of dive into and take a look at? We can kind of add annotations now. We've customized this, so now we can kind of log in and say, like, well, what changed on this day? Did we clean the instrument? Was there PM? Did we change the instrument, the standard lot that we're using, so we can kind of track what's changing over time with these annotations? And another cool feature is this Pareto plot visualization, where we can look at what is causing variability within my system. And so in this case, the retention time. So then for troubleshooting, we know, okay, we need to go mess with the LC and figure out why is it so variable and try and resolve that. But one of the next kind of step here, and kudos here to Vagisha, who's the lead developer that's enabled this, is that we've made this an automated process. So basically now on the instrument, they set it up and they define which samples are the QC samples, and they direct it, and it goes straight into our Panorama server. And so we can then walk up at any day, and we have this nice new view now as a QC summary, and we can walk up and log in the morning, and we can say, okay, here are all the instruments in the lab. These two instruments are actively running. This one, everything's green, everything's checking out. Now this one, there's something going on, and we want to go take a look at that particular instrument. So this new summary view has been really fantastic for us. And this is one of the biggest things for the program thus far. That's really every day to day that we're using it real time, all the time, and really depending upon. And at the front end, Skyline was originally developed for targeted proteomics, but it's expanded now to support small molecules. So we have to kind of, when we say we're now a targeted mass spec software, right? And so we've now expanded that within Panorama as well to support small molecule work. And so this is an example of a lipid panel, some lyso-PCs from some folks. And so that's kind of some features that we've been working on are kind of developed and we're kind of refining now. But now I'm going to kind of talk to you about some more of our features that are in development and kind of where we're still in the vision stage and trying to figure out how are we going to implement this on our server. So a lot of these pieces, the method build, refinement, data storage analysis, all of those features within LabKey, like you can take advantage of today on your own servers. They've been released. But now we're kind of in the, what about that other vision of we want to analyze our data, especially some of the routine things that you do every day within LabKey once the data is there. And beyond that, like in our hands, we have to, after we generate, kind of QC our data, we generate our nice summary table, say, where we have peptide or biomarker and concentration. We have to merge that back with clinical data, other biomarker data to kind of then guide decisions and figure out if what was the conclusion. And one way we're doing that is through a software which is Spotfire. And though there are a number of different solutions, this is one that our department is kind of piloting right now. So I'm going to first tell you a little bit about one of the kind of within the LabKey environment, one of the R packages that we're trying to adopt and get working on our server, which is Quasar. And Shadi is actually here today. So she has joined us about a year ago, not quite this year, earlier this year, and has made huge progress at implementing this on our server. And this is a visualization I'll walk through in a moment that she's kind of playing with now. But so what is Quasar? So Quasar was an R-script package that was developed at the Broad in Steve Carr's lab. And it was to support the multiple reaction monitoring or the multiplexed type data that I was generating by mass spec. And it does in an automated way. It actually helps you quickly calculate your limits of detection, your limits of quant for all of your peptides. It looks at the variability, so it's kind of basically that nice core statistics that you really need to do to QC your data at the front end. And one of the neat features that it has is this, what they call this audit algorithm. And it evaluates kind of the quality of your replicates at the transition level and helps you detect interference. And this is a visualization that Shadi did, ranking basically from peptides that I was screening that were performing well to peptides that weren't performing well. And so the darker, the more red, it was like the fewer transitions that I had that basically passed the QC criteria. And we were able, and this was like a calibration curve and then a few samples. So basically this particular peptide was doing really well, and then around here we're starting to get to where our LOD and LOQ are. And so this is kind of something that we're playing with now, but it's kind of a neat example of something that you can do and is enabled on this server, and it will be really powerful for us to be able to kind of quickly just glance through and get a sense of, how things are performing. And so the other piece of it is we're connecting our LabKey server to a Spotfire server. So it's now sourcing all of the data that we're inputting from the mass spec directly into Spotfire, where we have built some templates, as I said, where we're merging with some of our other data types. And so this is one new thing that we're doing was kind of complicated in-house to get this working, getting these servers talking to each other. But the LabKey team really helped us get this working. And so we're starting to play with this some in-house to kind of realize that whole kind of workflow now. So it will be a lot simpler and more trackable than it was at the earliest stage. So So I feel like this went by really quickly. So, just kind of back to the biomarker development pipeline, I hope kind of given you a picture of how we're using LabKey to help us with our assay development and kind of to manage our data, and how we're kind of working on this piece of it now, but it really is accelerating drug development and biomarker development within my group at Genentech. So I guess we'll welcome any questions that you guys have after. I thank a number of folks. A few folks are in this room today. Josh from LabKey and Brendan are here, and Shadi is also here with us. And then a number of other folks at Genentech, especially Laura and Josh, that have been helping us get this connected with Spotfire. And a shout out to my colleagues at Roche, Tom and Michelle, who really, we both came independently to this mass spec conference and saw a solution. And so it was kind of a unique case at our company where we're reaching across the pond to find a solution and get this working. And it's really, Panorama is really expanding use at Genentech now. So.